Hi, my name is Stephanie Strom, and I'm a student in the muscle biology lab at UMass Amherst. This presentation today is on the effects of aging and obesity on skeletal muscle fiber size and myosin heavy chain isoform distribution in humans. Just for some background on this project, in 2020, 988 million people aged over 5 years globally were classified as obese, correlating with a BMI greater than or equal to 30 kilograms per meter squared. And it's projected that in 2035, 1.9 billion people will have obesity according to the World Obesity Atlas. This is important because obesity itself is a risk factor in many life-threatening diseases such as cardiovascular disease, type 2 diabetes, and certain cancers. In addition, relevant to this project, it's related with impaired skeletal muscle function. As you can see in the figure on the right, when you have a lot of excess adipose tissue, as shown in Part B, it can surround all of the little myofibrils within the muscle fiber, and therefore that leads to an increase in a non-contractile substance because adipose tissue does not have contractile proteins in it, and therefore it can lead to the muscle producing less force. So it's theorized that muscle that is abundant in adipose tissue may not function as well as muscle not inhibited by excess fat. In addition, within these muscle fibers contains a protein known as myosin, which is a major contractile protein in muscle tissue in all three types of muscle tissue. It converts ATP into mechanical energy, which in turn produces force and movement. 3 isoforms are expressed in human skeletal tissue, one of which being MHC1, which is lower force, lower velocity, MHC2A, which is higher force, faster velocity, and MHC2X, which is higher force, fastest velocity. Type 1s are known as slow twitch, and type 2 are known as fast twitch. The study in particular that I am referencing is the R21 or MAT study, which stands for muscle adipose tissue. It's a cross-sectional study that involves an outcome status at one time point across specified populations. The study goals include analyzing intra and intermuscular fat content relative to the molecular, cellular, and whole muscle level function within different populations to determine a relationship between adiposity and skeletal muscle contractile properties. This study is being used for many other projects, but specifically I will be focusing on the muscle contractile properties, namely myosin heavy chain isoform distribution. So some participant characteristics that I'll show in the next slide. There's young normal BMI versus young high BMI versus older normal BMI, and it's also sex-specific into male or female. Here you can see some participant characteristics in the 3 subgroups, such as sex, age, height, body mass, BMI, moderate to vigorous physical activity per day, peak muscle cross-sectional area, and specific isometric torques. The participants were controlled to maintain a similar moderate to vigorous physical activity per day. Now onto the methods. There are 5 steps that I have used, one of which preservation of biopsy tissue, 2 tissue sectioning, 3 immunohistochemistry, 4 imaging, and 5 analysis. The first step is preservation of biopsy tissue, which is not done by myself. It's done by members outside of the muscle biology lab, and it's done immediately after the muscle biopsy with the tissue placed in Tissue Tech optimal cutting temperature, which is known as OCT, and then it was stored at negative 80 degrees Celsius. You can see the muscle tissue on the right embedded in the OCT. The muscle fiber itself is the red speck, and the OCT is the surrounding white compound. The next step is tissue sectioning, where I made 6 micron cross-sectional slices using the cryostat as shown above at negative 20 to negative 23 degrees Celsius. Tissue samples were secured in OCT, sliced with a razor blade, and adhered to glass slides. They were stored at 4 degrees Celsius for at least 4 hours. I improved the technique in this step by using the paintbrush method instead of using the glass anti-roll bar that usually flattens out the tissue. Instead, I would guide each tissue that's coming off the blade with a paintbrush so that it lays flat, and this prevents any additional tearing that would hinder the images. The next step is immunohistochemistry, which is using antibodies to target a specific protein of interest, and in this case, I'm using it to target the 3 myosin heavy chain isoforms expressed in human skeletal tissue. And the general steps include creating a hydrophobic border around the fibers with a mini pap pen, as you can see in the figure, incubate them with a primary antibody solution overnight, then you incubate them with a secondary antibody solution for 1 hour, which is what would provide the fluorescence so that I can actually see the myosin heavy chain isoform expressed in each fiber. Next, you would post-fix the samples with methanol, apply Vectis Shield, cover slips to then preserve fluorescence, and then you store it in the 4 degrees fridge until imaging. I should add that you should wash the samples with 1x phosphate buffered saline in between each step to produce the clearest results. And also, it's important to know that you need to incubate the samples in a humidified environment, as shown using paper towels in a cigar box so that the samples don't dry out overnight. The next step is imaging. Using the Kion's BZX810, the samples are imaged in 4x, 10x, and 20x, and I add a 100 micron scale to each photo before analysis. Here you can see in 20x examples of images that I have gotten throughout the past year. As you can see, the green fibers are MHC1s, the red fibers are MHC2As, and the purplish blue fibers are MHC2AX. MHC2X alone is rarely expressed in humans. It's usually expressed as a hybrid with 2As, so anything that you can see that is purplish blue, that isn't 100% red, is one of those hybrids. And I have 3 examples. I have a young normal weight male, a young obese female, and an older normal weight female. Here are some examples in 4x. As you can see, the fibers have all different types of shapes and sizes, and the colors of the fibers are slightly different depending on the image. Once those images are generated, I move on to analysis, and I use ImageJ or Fiji to analyze the images, which includes outlining each individual fiber in accordance with the MHC isoform, as you'll see in the next slide. Here are some examples of analyzed images. As you can see, every fiber has been outlined and labeled. I start out with the MHC1s, then I move on to the 2As, and then the 2AXs. Here are some more examples that are just a little bit more zoomed out. As you can see, fibers can have as little as 100 fibers, and some samples can have as many as 300. Once the image analysis is done, results were created, and these are 2 examples of age by weight bar graphs. One is the isoforms versus cross-sectional area, and one is the isoforms versus the percent composition of each isoform. The blue represents the young normal weight group, the pink represents the young obese group, and the green represents the older normal weight group. As you can see, when comparing within isoforms, the young normal weight group has significantly larger fibers in terms of cross-sectional areas when you compare it to both the young obese and the older normal weight. And you can see the same result in the MHC2A fibers. The MHC2X fibers, the young normal weight group has significantly larger fibers than the older normal weight group, and the young obese group has significantly larger fibers than the older normal weight group. And then once you get to percent composition, there was actually no significance between the 3 groups within the 3 isoforms. But in general, you can see here that the young normal weight groups tended to have larger cross-sectional areas. Histograms were also made to show the age by weight results, which is comparing the percent frequencies of myosin heavy chain isoforms and their cross-sectional areas among the 3 subgroups. So as you can see, the young normal weight tended to have a higher frequency of larger cross-sectional areas compared to the other 2 groups when you're looking at MHC1s and 2As. But then 2X, there's less of a obvious trend towards a higher cross-sectional area in the young normal weight group. When broken down by age, by weight, by sex, there were more interesting findings. As you can see across the three isoforms, men had significantly larger cross-sectional area averages within the fibers when looking at the young normal weight groups and the young obese groups. However, there was no significance between the two sexes in MHC2As and 2AXs when looking at the older normal weight group. Interestingly, females had a higher cross-sectional area in the older normal weight group of MHC1 fibers. In conclusion, age and weight reduce single fiber size in human skeletal muscle, potentially due to an atrophic response that could alter cell function as well. Sex also appeared to play an important role in the young because females had smaller fibers in the normal weight and obese cohorts, but there was also no fiber shift towards more slower contracting fibers observed with obesity or age, possibly due to matching groups for physical activity levels. This work is part of a larger study that collected information on skeletal muscle structure and contractile performance at the whole body, whole muscle, single fiber, and molecular levels in these three groups. The work will be added to the large data collected at various anatomical levels to provide a clearer understanding of the effects of age and obesity on human health. I'm just ending off by saying thank you to Dr. Mark Miller and Dr. Becky Miller for the support and guidance through this project. Thank you to all the other members of the Muscle Biology Lab for all the help throughout the past year, and I'm extending additional thanks to Dr. Stuart Chipkin and Dr. Jane Kent. Thank you so much for listening.